In this video, let's understand what is a vaccine, a basic of vaccine, how vaccine is developed, different approaches to develop a vaccine, what are the different vaccines which has been developed world over, what is difference between Covaxin and covishield, which is better, and also what is pandemics new to the world? No, pandemics are not new to the world. Let's understand one by one in this video. So first of all, let's understand what is a vaccine. Vaccine contains the same germs that cause disease. For example, measles vaccine contains measles virus and HIV vaccine contains HIV bacteria. But they have been either killed or weakened to the point that they don't make you sick. Some vaccine contains only a part of disease germ. So this is the way how corona vaccine will work. Scientists have taken genes for the spike protein and surface of coronavirus and put them into harmless virus to make a vaccine. This is injected into the patient. When the vaccine enters the cells, which then start to produce the corona spike protein, this promotes the immune system to produce antibodies and activate killer T cells to destroy the infected cells, right? If the patient encounters coronavirus again, the antibodies and T cells which have the memory will be triggered to fight the virus. This is the mechanism which we can see when the vaccine is induced into the human body, right? So as I already said, a vaccine stimulates your immune system to produce antibodies exactly like it would you exposed to the disease. After getting vaccinated, you develop immunity to that disease without having to get the disease first. This is what makes vaccines such a powerful medicine. Unlike most medicines which treat acute diseases, vaccines prevent them. Rather than treat acute diseases, vaccines are going to prevent them rather than treat a cure. How does a vaccine works then? Vaccine reduces risk of getting a disease by working with your body's natural defense to build protection. When you get a vaccine, your immune system responds. It recognizes the invading germs such as the virus or bacteria, right? So this is how a vaccine works. A weak or dead form of germ is introduced into a body. That's what vaccine is all about, as we already discussed. When this weak or dead germ is introduced into your body, this sparks your immune response to develop antibodies, and as well as they remember the germ. The process itself is remembered by your cells as well as the T cells. So it generates the antibodies to fight the germs which invades the body, right? So this is whenever the new COVID virus enters into a body, when you are vaccinated, what happens is, since your body has already remembered, your cells already remember the way to destroy the new antigen which enters into a body in terms of COVID infection. So it actually develops an antibodies and kills it, right? So it produces antibodies. Antibodies are nothing but proteins produced naturally by the immune system to fight the disease. It remembers the disease and how to fight it, as well as if you are then exposed to the germ in the future, your immune system can quickly destroy it before you become unwell. That's what it makes the vaccine superior than the medicine. How? Medicines will be a cure when you are infected with a disease, where in case of a vaccine, before you are infected with, before a virus enters into a body, it fights back with your antibodies, right? So the vaccine is not therefore a safe and clever way to produce an immune response in the body without causing illness. Our immune system are designed to remember. Once exposed to one or more doses of vaccines, we typically remain protected against a disease for years, decades, or even for a lifetime. This is what makes vaccines so effective. Rather than treating a disease, as I already said, after it occurs, vaccine prevents us in the first instance from getting sick itself. So now let's understand what are the different types of vaccines which has been developed all over the world for COVID-19 especially. since the world over COVID-19 was a pandemic, the research was going on world over, so there were almost 200 vaccine candidates for COVID-19 being developed. Of this, 52 candidates of vaccines have been gone for human trials, right? So 52. The different types of vaccine in development of different types of vaccines, there are 3 different approaches, 3, 4 or 5 different approaches where vaccines can be developed. So let's understand one by one what are the different approaches where the vaccines can be developed. First one is the whole virus or bacteria method. The whole virus has been taken in order to develop a vaccine. Now the next is just the parts of the germs that triggers the immune system has been taken. For example, in coronavirus, the spikes which are also a protein has been taken or extracted in order to develop a vaccine, right? And the third type, the genetic material that provides the instruction for making specific proteins and not the whole virus, DNA or mRNA. In case of COVID, it's mRNA, which is a genetic material for COVID-19 or SARS-CoV-19, what we call as. So that will be extracted in order to make a vaccine, right? So these are the this is a genetic code vaccine where DNA or RNA, a single helix, as you know, the DNA is a double helix genetic material, whereas when it comes to RNA, ribose nucleic acid, it's a single helix genetic code vaccine, right? Viral vector vaccine. In viral vector vaccine, what we do is a vector has been selected. It might be a vector, it might be a which is congenial to human body, congenial to human existence. Those kind of germs have been selected, and that will be injected with the COVID material, and these viral vector vaccines are also being developed. So those are what AstraZeneca, Oxford, J&J, Casino and Gamaleya are viral vector vaccines, whereas genetic code vaccines are Pfizer, BioNTech and Moderna. So what are these subunit vaccines? Novavax has developed a subunit vaccines. For example, in corona, you can see these spikes, right? These spikes are nothing but proteins. These proteins are extracted, and this will be used in a vaccine to respond for our human antibodies, right? So this is a subunit vaccine, that is Novavax. A weakened or inactivated vaccine, where whole virus has been taken, whole virus of corona, in this case, has been taken. It's been inactivated or weakened. It may be killed by use of chemicals or the heat, or it will be weakened by using some chemicals so that it won't cause the diseases in the human body, right? So this is a whole microbe approach where the whole microbe has been taken, whole virus has been taken. So this is an inactivated vaccine. This is live attenuated vaccine. These are viral vector vaccine. Now what is the difference between inactivated vaccine and live attenuated vaccine? In inactivated vaccine, the virus is killed, whereas in live attenuated vaccine, the virus will be weakened. That's it, right? So let's understand one by one what is the whole microbe approach. As we already discussed, in whole microbe approach, the whole virus, the whole coronavirus is being taken as a vaccine rather than the subunit, a protein, not like a protein or vector. The whole virus, the whole coronavirus has been taken in order to instigate our antibodies, right? So the whole virus, if we inject a whole virus, definitely it will cause the disease. But in whole microbe approach in vaccines, what we do, we either inactivate it or weaken it. So how do we inactivate it? The first way to make a vaccine is to take the disease carrying virus or bacteria or one or very similar to it and inactivate or kill it using chemicals, heat or radiation. This approach uses the technology that's been proven to work in people. Excuse me. This is the way the flu and polio vaccines are made. The flu and polio vaccines are made in an inactivated whole virus approach. and vaccines can be manufactured on a reasonable scale, not on a large scale, but on a reasonable scale, right? However, it requires special laboratory facilities to grow the virus or bacterium safely, can have relatively long production time. So this is a drawback, a relatively long production time, and will likely require 2 or 3 doses to be administered. This is a disadvantage in whole microbe approach and specifically inactivated approach. So now let's look at live attenuated vaccines. A live attenuated vaccine uses a living but weakened version of the virus, or one that's very similar. Measles, mumps, or rubella vaccines and chickenpox and shingle vaccines are examples of live attenuated vaccines, right? These are examples for live attenuated vaccines. So next one is a viral vector vaccine. In viral vector vaccine, what we do, we use one of the bacteria or virus as a vector. Vector is nothing but a carrying agent, that's it. It carries a genetic material or virus. In that case, in order to make it too simple, Aedes aegypti is a mosquito which carries malaria as a vector. Mosquito Aedes aegypti, if mosquito bites you, that it doesn't causes malaria for you, but it carries the malaria disease, right? So this is a vector. In the same case, in viral vector vaccines, a vector has been used. It might be some symbiotic bacteria or a virus. So in that, the actual virus will be injected, right? This type of vaccine uses a safe virus to deliver a specific subparts called proteins of the germs of interest, so that it can trigger a immune response without causing a disease. It doesn't cause a disease, but it will trigger your immune response, right? Instruction for making particular parts of the pathogen of interest are inserted into a safe virus. The safe virus then serves as a platform. It serves as a platform or a vector to deliver the protein into the body. The protein triggers the immune system, so then antibodies will be released, so the invading virus will be killed, right? The Ebola vaccine is a viral vector vaccine, and this type can be developed rapidly. As you know, Ebola was also a deadly disease, but it didn't turn into a pandemic. It was curtailed in Africa and South America only. What happened with Ebola? Ebola was really contagious. It was spreading easily, but again it started mutating. When the world over, it was really a concern. It started mutating. The Ebola lost its contagious ability, the spreading ability, but it became deadly. But later it was curtailed. Now it's been Ebola is not such a threat as like COVID-19, right? The Ebola vaccine is a viral vector vaccine that this can be developed rapidly, right? So the other approach is a subunit approach. Rather than the whole microbe approach, the subunit approach is somewhat different. A subunit vaccine is one that only uses a very specific part of a virus or bacterium that immune system needs to recognize. It doesn't use the whole virus itself. It doesn't take the whole virus itself. It only uses a part of a virus. It might be a spike protein, or it might be any part of a virus, right? So it doesn't contain the whole microbe or use a safe virus as a vector. It doesn't use a safe virus as a vector also. The only thing is a part of a subunit. You should understand the difference between viral vector vaccine as well as subunit vaccine. In a viral vector vaccine, what we do, we use a virus or a bacteria as a carrying agent, but in the case of subunit, we don't use a viral vector itself. We only take a subunit of the whole virus, right? So the subunits may be proteins or sugar, it should not be a protein. So most of the vaccine on the childhood schedule are subunit vaccines protecting from peoples that has whooping cough, tetanus, diphtheria. As you know, DPT is a very famous and very useful vaccine which is been vaccinated for the child, right? Diphtheria, pertussis and tetanus. Pertussis is nothing but whooping cough, right? So this mechanism of subunit approach is used in diphtheria, whooping cough, tetanus and meningococcus meningitis as well, right? So next way of developing a vaccine is the genetic approach or nucleic acid vaccine. What we do in subunit approach, we used to take a part of a virus or bacteria or an organism. It might be a protein or whatever it might be, but in genetic approach what we do is we extract the genetic material. As we already know what genetic material is, either it might be DNA, deoxyribonucleic acid or RNA. DNA or RNA will be extracted in nucleic acid vaccine. Unlike vaccine approaches, thus either weakened or dead whole microbe or parts of one, a nucleic acid vaccine just uses a section of genetic material that provides the instruction for specific proteins, not the whole microbe. DNA and RNA are the instruction our cell uses. As you know, the DNA or RNA are the genetic material as the cell uses. So it makes proteins in our cells. DNA is first turned into mRNA or messenger RNA, which is then used as a blueprint to make a specific proteins, right? So this is how a genetic approach, a nucleic acid vaccine is developed, right? So this is a single helix, as you can see, single helix. So single helix means it will be a RNA based vaccine, right? So this is DNA, which is a double helix matrix. Since you guys are preparing for other exams, you should be knowing who was the major brain behind Watson and DNA, the Watson and Crick and all. So then it will be transcribed into RNA transcription, and this will be dealt in a different video. So DNA will be transcribed into messenger RNA, right? A nucleic acid vaccine delivers a specific set of instruction to our cells, either as a DNA or mRNA, for them to make this specific protein that we want our immune system to recognize and respond to, right? So this is how DNA based or genetic approach based vaccine is been developed. So now in this slide, let's understand, as we already discussed, type of corona vaccine approaches. So DNA and RNA based approaches, and live attenuated based approach to develop a vaccine, inactivated based developed to approach a vaccine, subunit based, either the proteins or some other materials will be extracted from a vaccine and extracted from a microorganism, and it will be developed as a vaccine. And the viral vector vaccine, as we already discussed, a congenial egg, a virus will be used as a vector, a carrying agent, where the other disease causing virus will be injected into it, right? So how it works, advantages, disadvantages, existing samples, group testing for these in approaching in COVID-19 and all, we already discussed about this in the previous slide in detail, right? So please have a look about it. DNA and RNA, the vaccine uses DNA or RNA molecules to reach the immune system to target the key viral proteins. In case of live attenuated, this is a weakened version of the actual virus. In case of inactivated vaccine uses the whole virus after it has been killed with the ETA chemicals. In subunit approach, what we do, this vaccine uses a piece of virus surface to focus your immune system on a single target, right? In a viral vector vaccine, this approach takes a harmless virus and uses it to deliver a viral gene to build a immunity, right? So this thing we already discussed in detail, right? So different COVID vaccines which has been developed in the world. As of now, you know, COVID was a serious threat from 2020, 2019, 20 and all. So the whole world, whole pharma sector was behind it in our development of the vaccines. So in this quest of developing a vaccine, most of the pharma companies have come up with as we already discussed. 200 were trying for it, but 50 to 52 companies have been granted with the COVID vaccine, and they went for human trials as well. After that, there were very finger counting numbers which has been developed, right? So how some of the COVID-19 vaccines. So these are the COVID-19 vaccines which has been given approval from the respective national governments. It might be US, or in case of India it might be DGCA. In case of US it might be their own drug control authorities, right? In case of Russia, France and all, they have developed their own vaccines. So first one is Oxford Uni AstraZeneca vaccine, which has been developed in Britain. It's a viral vector genetically modified virus vaccine. So it should be the jab should be in 2 doses. You should be take two doses, and it should be stored in a temperature of 2 to 8 degree centigrade. So the duration between the 2 doses should be based on the experts, right? So Moderna is a US based company. It has developed a vaccine, Moderna vaccine. It is a RNA part of virus genetic code, and again it is also a 2 doses vaccine. The storage capacity, the storage temperature is -25 to -15 degree centigrade. Pfizer and Biotech, which has been developed in collaboration with US and Germany. It is a RNA based vaccine. Again, it should be taken in a 2 doses. It should be stored at a temperature of -80 to -60 degrees centigrade. Gamaleya or Sputnik-v, which is a Russian based vaccine, which is recently launched in India as well. It is a viral vector vaccine. It should be taken in 2 doses, but now they have been changed it to single dose vaccine. It's a viral vector vaccine. It should be stored under -15.5 degree centigrade, right? Sinovac or Coronavac, which has been developed in China, is an inactivated virus weakened vaccine, and it should be taken in 2 doses. It should be stored in a temperature range of 2 to 8 degrees centigrade. Novavax, again developed by US, it's a protein based vaccine, should be taken in 2 doses. The jab should be taken in 2 doses. It should be stored at 2 to 8 degrees centigrade. In the same way, Janssen as well, it's a viral vector vaccine, right? In India, there are 2 vaccines which has been developed and which has been tested, and as we know, which is been taken by Indians as well. Those are Covishield and Covaxin. Let's let's understand the difference between Covishield and Covaxin. Which are the companies which has been developed, which is the company which has developed Covishield as well as Covaxin. What is the efficacy rate? How different are they, and which is the best among them, right? Covishield is a Oxford AstraZeneca vaccine being manufactured by the Serum Institute of India. It's made from a weakened version of a common cold virus known as a adenovirus from chimpanzees, right? It has been modified to look more like a coronavirus, although it can cause illness. How it works: when the patient gets a jab of a vaccine, it stirs the immune system to start producing antibodies and prepare it to attack any coronavirus infection, right? Efficacy: Covishield has over efficacy of 70%. However, it can be over 90% when it administered as half dose followed by a full dose a month later, which is in the news in recent times. We will discuss it in the future videos, right? So it should be stored safely stored at a temperature of 2 to 8 degrees Celsius, which is very much important. Whereas in case of AstraZeneca based vaccines and all, it should be stored under -degree centigrade, whereas this is good for Indian thing, right? So this is the price which has been fixed, and later it's been revised as well, right? Covaxin, it's an inactivated vaccine, which means that it is made up of killed coronavirus, making it safe to be injected into the body. Covaxin has been developed by Indian biotechnology company Bharat Biotech and clinical research body ICMR, right? Bharat Biotech used a sample of coronavirus isolated by INMV, Indian National Institute of Virology. When administered, immune cells can still recognize the dead virus, promoting the immune system to make antibodies against the pandemic virus. How it works: and delivery of the vaccine teaches the immune system to make antibodies against the SARS-CoV-2 coronavirus, right? So when it recognizes the SARS-CoV-2 vaccine, when we are infected with the SARS-CoV-2, then the remembered memory will be activated, antibodies will be activated to kill the new corona, right? So let's understand the efficacy. Covaxin has shown a 78% efficacy, whereas in case of Covishield it was 70% efficacy. In second interim analysis, 100% against severe COVID-19 diseases, right? Storage. The vaccine can be stored at 2 to 8 degrees Celsius, right? So, which is best, either Covaxin or Covishield? Don't look for which is best in terms of efficacy and the clinical trials. Both vaccines have been yielded a good results. So, go for vaccination. That is at most importance, right? So, mutation of coronaviruses in India. What is mutation? Mutation in simple terms is when a virus enters into a body, it enters into a cell, it reproduces or replicates into multiple copies. During this replication of copies, the basic genetical coding of the virus will be changed, right, in a minor way. In the process of the multiplication of copies, the virus itself, the genetic coding will be changed and the new variant will be formed. This is what mutation of coronavirus is all about. So there are different genes, different variants of coronavirus genes which has been found world over. N501Y, which has been found in UK, South Africa and Brazil. This mutation is located in the viral reception binding domain for cell entry, helps the virus to bind more tightly to the human cells. So E484K is one more mutation which has been found in again Africa, Brazil and US. E484Q is found in South Africa and Brazil. E484Q plus L452R. These are only the names which has been given to the variants. In the same way, in India also there is B series of variants which has been found, and the scientists are opined that the Indian variants are more deadly compared to other mutated viruses. So mutation will be going on. It might be for coronavirus, it might be rhinovirus, or for any other virus. The mutation will be going on. Even in Ebola also, we had seen that the mutation will be going on, right? So as mutation will be going on, we should understand, since we are going for a vaccination, does the vaccination, either it might be Covaxin, Covishield or Sputnik, will it be helpful for the mutation or not? So what ICMR, Indian Council of Medical Research, announced that Covaxin has neutralized multiple variants of SARS-CoV-2, including the double mutant strain called B1617, right? So in that case, even the virus mutates, but the vaccine which we are taking will be really helpful, right? So this thing which we already discussed about the different types of vaccines which are compared, right? So this is all about vaccine and different types of vaccines which has been developed world over, and how vaccines are developed, what is the mechanism behind development of virus, how it will help us to fight the disease of coronavirus. Thank you. Please subscribe to Siddaganga Academy YouTube channel for more such kind of videos, right? Thank you.